things are going much smoother now. I see we meet regularly at 10 o'clock to have a short discussion on what kind of data we will take. And as of today, at least we took already magnetic field 3D and free motion. Thermal wasn't quite ready because nobody was in here this morning. But then we started to turn on the basement, B floor, 2D one tracking plus phototaxis is nearly ready, except tracking isn't quite working. So case by case, I think we are almost ready to start serious science data. And while we are waiting, stop, let me just remind you. I want to keep kind of a very quick daily meeting. Thank you for Richard and Ed and Pat coming at 10 o'clock. We have a good idea who need a sample. The quality of a sample is much better now. I can see a healthy moving shielding us from every experiment. So it's going to work very well. Microscope, we still have to do fine tuning. Joe is supposed to come back tomorrow. Anthony is leaving this weekend. Thank you for working hard. So next 2 days, I want to finish 9 confocal and 2D one tracker and turn on the 3D system. So it's coming along. The B floor by Friday should be ready to do both 2D and 3D. That's good. Unfortunately, power supply for electricity is kind of dead. We tried to the different one, may or may not work. So if the power supply isn't quite working up to 600 volts, maybe as we discussed A floor, we are going to convert to temperature business depending on the this new cooler systems. We don't know yet. All right, that's the kind of stuff quickly. Presentations, I should do it. Today we have a series of talks about optogenetics, which is a completely new territory. We haven't started yet, but I was thinking for 2 years, but didn't have enough manpower. But finally, we have enough people to think about the biology side and how to make a hardware. So 4 speakers today in a few minutes, just to let you know. It's exciting. I think it's going to work this time. We have enough manpower. Now, one more slide. As you know, we have a kind of a super group, B plus free motion, and E plus T, and downstairs virtual reality in 3D phototaxis like that. I think people begin to be equally distributed to find a good place to be. When you come in the morning, it's easier to talk to you in person and assign you one by one to a project which has no manpower. So come early in the morning if you want to have your own project. This is important. After this meeting, it's chaotic. I can't talk everyone person by person anymore, right? And as you know, we have 3 groups. So this is what I want to do and see how it works. Starting next week, you should present your status and plan, group by group, for the given date every week. So this is the kind of arrangement, looking at your available time slot. So I came up with the idea that Monday we start from technical side of status report, microscope, biological sample, in particular new technology, like how to make a 3D with electric field like that, or any new idea how to make a new mutant, a status mutant, or how to make an optogenetic sample like that. I think we want to cover a new technology and new microscope so that everyone is aware of what's going to happen in the future. Okay. Likewise, Wednesday I assigned all kinds of software guide to make presentations of status of especially new simulations. We are developing a complete simulation of 302 neurons communicating each other and making a lot of progress. I think we have maybe 5 or 6 full-time students working. 3 Chinese and Yushun and Ahis and also Yugo. So 6 people are working full-time for modeling. It's a huge manpower for next 2 months. Right? And then Tuesday, electric field and temperature share the same kind of system as I said. Once we finish Canon 60 based behavior experiment upstairs, we move to A floor next. And indeed, it's already happening for electric field since we don't have enough people and systems. Idea is to measure a damped sine wave and look at ASJ. We start to see ASJ and look at the correlation between the direction of the head and ASJ. So it's exciting. But the system gets more complex, so you need more experienced person to operate. Likewise, temperature, once we establish how to take the data as thermal behavior and hopefully 90-degree turn upstairs, we move to A floor because we have ASJ And we already received like one mutant of SJ with AFD and AIY and so on. So it's exciting. So finally, if we're lucky, in a couple of weeks, we are beginning to see a flashing neurons, at least 1 or 2 neurons under electric field and temperature. Okay, so that's E plus T group. So exciting. Let's finish Canon 60 data quickly and move to A4. Thursday is actually B flow activity. So we are turning on already for the taxis with 2D, and Anthony and also Javier is tuning line confocal, which may take 2 weeks. So as I said, we work on line confocal tuning in the morning. After 3 p.m., we devote the system to phototaxis experiment every day from now. Okay, and 3D, as I said, Joe is coming back tomorrow, and we start to use 3 centimeter. And now electric field for 3 centimeter is ready. Who's preparing at this period?
Andrew.
Andrew.
And Rachel.
Rachel. So we may try already a 3D 3 centimeter cube with electric field tomorrow or Friday. That's exciting. If you see this cone shape, that is a kind of very, very impressive measurement. Any observation with 3D, with stimulation, nobody has ever achieved. Okay, so this is exciting. One idea. And Angela, and B field seems to be Friday's convenient. Am I correct?
Well, yes.
Hello, is that correct?
Yes.
So I assigned free motion and B field to present every Friday. Make sense? Now it's up to you. I just typed some senior member and active member, but within a group you should define a coordinator. And you have about, say, 30-minute presentation, plus some questions. So if you don't have much to say, you should cover some interesting paper. Okay, you have an option. Either you present an exciting new observation of yourself, or present important paper, or come up with a new idea what to do in the future in a group every week. Do you agree? Let's make sure we have equally distributed members for each science group. E plus T, raise your hand. Roughly speaking, electric field. Raise your hand. 1, 2, 3, 4, 5, 6. Okay. P3D, you are also part of that. And this is a new group. You should be a part of that. Harrison, you should be a part of that, maybe. Who want to go B floor, interesting in more microscope hardware and operate 2D, 3D? But you have to have a laser safety. Raise your hand. I think it's ready next week. And we already, Harrison and Rachel are downstairs today, and we need more people. We can have 4 or 5 people, just to let you know. You're welcome to shift if your group is too busy or crowded. I may have a lot of students actually. This B plus free motion, raise your hand. We have a lot of free motions. That's okay. So it's reasonably equally distributed. So whoever senior member should try to head count and make sure who is going to say what. If nobody is willing to say, you have to say something. Right? So I want to reduce my organization of my lecture at minimum as possible starting next week. And tomorrow I give a lecture one way or another, maybe origin of life again, and then I'll finish my lecture. Questions? Make sense? Do you understand? Everyone know what you want to do, and you know who to talk after this meeting. So why don't you have a short meeting, group, and take a data? This schedule works for you, more or less? Okay, and then let's start today's presentation. As I said, so maybe I show a few slides as an introduction. Optogenetics, you're going to hear a lot of talks, 4 talks. But basic principle, maybe I show this slide, is to understand signal propagation from 302 neurons to the next time. And just to remind you, my concept is that even in a brain, every single neuron has to receive a signal from previous time and send the signal to the next time. So let's analyze in 4 dimensions. So as a function of time, when the sensory signal receives a signal, it goes to interneuron and it goes to motor neuron, according to the conventional picture. What we're trying to do is to prove a new concept that any sensory signal only carry information in time, so you have to tell my brain how I am moving as a feedback from motor neuron. By combining these two, you can perceive a space, and the easiest way is to have a clock by central pattern generator. So you are clocking, so you are measuring a temperature, left, right, and I know how fast I'm going. So by combining my own motion and signal in time and clock, you are going to map out the temperature in space. That's the idea we want to prove, just to let you know. And this goes into your brain as a memory, and this memory compare with the next signal and next motion, make sure everything is consistent. And that sensation, I claim, is the origin of perception of the space and become eventually consciousness. Now, how am I going to study it? Basically, the CLNS has roughly sensory neurons, interneuron, and motor neuron connected like this. So what we want to do is to, as I said, signal from here to here is a time domain, here to here is a space information. And we want to study this by watching the brain, and how it works is by this map. So eventually, in a few weeks, we are going to see 302 neurons, 30 times per second. Nobody has ever seen it. The previous observation, just to make sure, was only one three-dimensional volume per second. And that is too slow. So we are speeding up the microscope so that we can observe all 302 neurons 10 times or 20 times or maybe 30 times per second. And that allows us to look at clock in the brain, typically 1 Hz, and how they coordinate. Signal from here to here may take a fraction of the second. But if you are sampling every 1 second, it's too slow. Signal from here to here may take only 10 milliseconds. And then, in order to see a speed of signal propagation, of course, you have to measure 100 times per second like that in 3D. And that is the microscope we are designing. And at the same time, if you want to follow this step by step, best way is to have a kind of connection and simulate. And of course, I showed many times. Basically, 302 neurons are connected to nearby neuron by this matrix, connectome. And it consists of gap junction and two different types of synapse, excitatory or inhibitory by this. And I said this is the sensory to interneuron. Signal from sensor go to interneuron, and interneuron to motor neuron here to here. But feedback from motor to interneuron comes here, and that basically go back to central interneuron and perceive a space here somewhere. At the same time, I said there is some kind of reflex, like very serious avoidance behavior. Say, when I see a snake, I go backward. Immediately it comes from sensory to motor part. Skip interpret like that, right? And there's also internal connection between sensor neuron and sensor neuron, like a H finder in my eyes, or my body motion for sine wave is the communication locally. This is the feedback. This kind of pattern, like my working at constant speed, moving this pattern, I don't have to use my brain. It is already hardwired to make sure everything internally in the motor neuron, and I can find the H by communication here. So this has a lot of information. The next is how to simulate this. And unfortunately, what's missing is whether this synapse is excitatory or inhibitory. That information does not exist yet. So how can we measure it is a real question. So that is my major motivation to go into optogenetics you're going to hear. Optogenetics is basically, ideally, you basically shoot a light into even interneuron, because interneuron you genetically code as a photoreceptor. You shoot a light, this is activated. Once this neuron is activated, it's going to propagate to this line. Basically, you have this. So anywhere in this map, let's shoot a beam here. And then you see a propagation of this signal to the next stage. And then you can map out this by yourself purely by optical measurement. And you can even tell whether signal propagation is positive or negative. And then we can improve this map and put into the computer simulation this guy are making, and that's it. This is the end of the story. If we can do it, basically it's like a human genome project. This is a C. elegans dynamic brain project, Connectome. Right? There's a big difference. Connectome, already published 10 years ago, is a static image. What we are going to establish is a connectome of dynamic brain. And why you are doing basically a behavior experiment? Because of course we are doing this at the black box. You basically shoot some sensory signal, you observe the motion. All the experiment upstairs by Canon is basically treat this at the black box. You only observe signal and motion. But then going to A floor, B floor, you are going to start to watch a human neuron to begin with. And in a couple of weeks, if you're lucky, we begin to see a 300 neurons. So by end of the summer, if we are lucky, we see all the neuron flashing, we have a computer simulation, and begin to do optogenetics. That's the idea. All right, so let's go optogenetics. You are first?
No, Charles will be
Huh?
first.
Oh, we start from hardware. I do not. So we start from hardware side, how to observe it, and then go to more detailed biology. Just to keep it. So Charles, I asked Charles, he was here already last summer, started to design a 3D system, so I thought it's a good project for him to think about hardware design.
Okay. Hi. So I'm Charles, and so I wasn't expecting to go first. And so if you have any question concerning biology, just ask, and Mikey and Michelle will answer your questions. So I will mostly present the paper from this guy, and we will see what we can learn from their system and how we can apply to our system. So this paper is simultaneous optogenetics manipulation and calcium imaging with C. elegans. It was published in Frontier in Neurocircuits in March 2014. So the main problem in optogenetics is how to target the neuron that you want to activate. So, because, for example, let's say you want to activate a mechanosensory neuron, for example, this one, AAVM, when you express channel rhodopsin in this neuron, all these other mechanosensory neurons will also express this kind of channel rhodopsin. So you want to target, for example, if you want to activate this neuron specifically, then you will need to target your laser specifically at this area of the worm. And the way you can achieve this is using a DMD, digital micrometer device, which is basically a lot of very micro mirrors. That
I think he's going to talk about it.
Yeah, so Jumbo is going to talk more about this in detail. So basically they rotate, and when you shine the laser on it, you can form any pattern that can allow you to, for example, shoot the pattern in here and here, or both at the same time. So this is the system that they have, and this is a combination of 3 systems. So behavior imaging, which is like our work tracking, fluorescence imaging, and then optogenetics. And so let's start from the easy part, so behavior imaging, which is everything here. And then, so the worm is on Eiger, an XY motorized stage, and they use dark field NIR light illumination. And they detect the worm using this high-speed camera. And this high-speed camera is linked to a computer software that will control the XY stage for worm tracking, and also
Do they use just 10x from the bottom? I don't know that.
yeah. So they only use this objective, the whole system.
So fluorescence image is not that high resolution.
No, you will see later like this. Not that
I
high.
think they improved their setup in a later.
They
I
have
remember
project-
seeing paper with
But
that.
yeah, I looked it up, but I don't think that setup has optogenetics.
No, it doesn't have optogenetics though.
Alright.
Ah, okay. They have 2 different systems.
Yeah.
With that optogenetics, they have 40x from the bottom, 10x from the top. That's what
Yeah.
your original design, right? When they do optogenetics, they combine one objective.
Yeah.
Oh, maybe our system is better then.
Yeah. So,
I see.
so, oh yeah. So this camera was is going to link to a software which is going to control the XY motor stage and the DMD. I will talk more about the software later. And then, so next for fluorescence imaging. So first thing I want to talk is that this DMD is used for optogenetics and the imaging system.
For imaging?
Yeah.
Why do you say imaging?
So, so basically, they will want to eliminate some specific gene.
For calcium imaging?
Yeah, for calcium
Oh, they don't shine the whole space?
imaging. No, they don't shine the whole space.
Why
They
is that?
use this DMD to also shine on specific neurons to eliminate the
Why
neurons.
is that?
So I'm not so sure about
So
this,
they
but
don't-
Is
Oh,
that-
so they only allow specific neurons for calcium imaging?
yeah. So for
So they
example,
shine a specific neuron for optogenetics, and the next
Yeah.
time they shift to G-camp.
so the thing is, so channel rhodopsin and GCAM are both sensitive to 473 nanometer. So that's why you don't want to, yeah. So if you want to see the activity of interneuron, for example, then you don't want to shine 473 on the whole thing because you will also activate the channel rhodopsin from other neurons. So that's why you have to be specific in where you eliminate.
Oh, so you use a DMD to like selectively eliminate both the.
You eliminate the neuron that you want to investigate, and you use it also for optogenetics to
Now,
activate.
if you have a line confocal, the position of the beam is changing at the function of time. So can we mix the same
So,
wavelengths if we have a line confocal in detection site? What do you think?
so for
Like this. It's better than this system, right?
yeah.
Here, fluorescence is a wide field, so they have to shine the spotlight. In our case, we are scanning a line at function of time, so given time, I know where we are looking at.
Yeah.
So this may be interesting project. A new idea. Separating time, because time confocal is scanning a field.
I mean, we don't have to scan the entire. We can also selectively like scan like region of interest, so that we
But
don't
if
look.
you have something like QW1217 to watch 100 neurons flashing, you have to scan more in time. Anyway, it's not that advanced system then.
Not that advanced. Yeah.
So just to make sure everyone, what we have downstairs, Javier design is we do have 10x and 40x. 10x is from the bottom for
Actually, I
behavior
have
observation.
the
This
picture.
part is the same. But sorry, you have our system. Okay. Yeah, why don't you explain the difference there? So this part is the same. But this part is flipped.
Yeah, so okay, so this part will be for this line confocal part, which is basically what we'll be doing is basically what their fluorescence imaging will be doing. So for our system, we might need the 561 nanometer laser put in here to.
We are adding 532 because
Yeah.
562 is expensive.
Okay.
And it's coming in a few days. And we have to have a dual view of 2 cameras in a week. So talk to, should we get dual view from WView from CMSI? Or get another camera? Whichever. Whichever is fine. Drone is coming in.
Or we just take.
Easiest way is to take out WView from CMSI.
The open spin, right?
Or we take the board from the dissection system.
That's okay, yeah, we are not using that board. So why don't you, did you pass dichroic?
Yeah.
The detection?
I think I need a dichroic, a different dichroic for that, right above the 40x, because we have 488 and 532. But
Did you get a machine filters?
we have,
All right, it's too technical, but we have
Yeah.
far more sophisticated systems than their system.
Yeah, so
And this
this-
paper was 2014. It's relatively new.
Yeah, so basically this part is the same as.
So
this
now I'm
part,
convinced, if we develop this system, it has 2 advantages. One is detection of fluorescence image is line confocal. So it's more three-dimensional and sharper image. And it's 40x with N0.8. It's much better. Second, our system has stimulation, blue light for 5 nanometer or infrared beam or temperature plate. So I think our system is as much as we can go, putting all existing technology in the flat geometry. Interesting.
Okay, so oh, okay, yeah. So just for a small recap, so the 473 nanometer will be used for both optogenetics and fluorescence imaging. For fluorescence imaging, it will excite GCOM3 to see neural activity. For optogenetics, it will activate channel rhodopsins and activate.
They don't use heterodopsin for inhibitor.
No.
What's the purpose of 561? That's for M5.
561 is for M-cherry. Yeah, so it will. Yeah,
Okay, so they just have a
just
channel.
as a fluorescence reference. Yeah. So, okay, the computer's software. So it's written in C. It's open source. You can look it up at this website. And so what it basically does, so it gets information from this camera, and it does worm tracking. We have that. And also it extracts the outline of the worm. And this is particularly important for their experiment because so in normal optogenetics, the cell, the neuron that you want to activate, you can target with, for example, M-cherry or RFP. But here, instead, what they're doing is they estimate the location of the neuron based on the shape of the worm. So, for example, in here, if, for example, you want to activate AVM, you know that it's basically around this part and more at the ventral side of the worm. So you, then they will shoot an illumination pattern at this side. So, and then once they extract the outline of the worm and they know where approximately they want to shoot, they send the instruction to the MD that will change the rotation of their mirrors. And then, so the speed of the software is very important for what is called DMD latency, because so DMD latency is how fast can the mirrors move after the worm move. So of course you want the mirror to move as fast as they can, and then they could measure. So basically, the latency between the two is about 28.6 milliseconds. So it's pretty good. So that means that the software is also very fast. And then so this latency, along with other system, I would say, other system error and stuff like. I mean, for example, the XY latency, modern state latency, the speed of the worm and stuff like this. It will all determine how close to target neuron they can be if you want to activate them separately.
Now, in neighboring, separation between two neurons are much shorter than 50 micron, I think.
Yeah.
So if you mark channel rhodopsin everywhere, it adds dense like 10 micron space. So 50 micron is not good enough.
Yeah.
So,
So 50.
Why do you say 50 micron?
So yeah, so 50. So because it has to kind of do with how they create their illumination pattern. So they kind of expect the neuron to be there instead of tagging it with an RFP. So that's why.
So here is 50 micron neuron this side, right?
Yeah.
So if you have marked channel rhodopsin everywhere, 50 micron includes maybe 5, 10 neurons within it.
Yeah. So basically, for example, if you
This one is separated. That's why it's easy. Yeah.
Yeah.
Why people publish the paper based on this neuron? It's so separated. Motor neurons. Easy. It's motor neuron. You shoot it anywhere here. Of course, it works. Go to the left and right. It's like a robot. What I want to do is to shoot somewhere here. It's dense.
We can actually use the top camera to actually locate the neuron itself to a higher resolution. And
Hopefully. Otherwise, I think we are talking more advanced technique.
But I think-
most of the paper is motor neuron.
Yeah, but then like
Kind
you
of
need
hard,
to
but
have like,
of course it works.
but I think it's like even faster update because like you have to like micron level accuracy.
Yeah, it's difficult.
Yeah.
That's what we are shooting. It's a challenging project now. You know. This paper is all about motor neurons.
Yeah.
Well, sensory neuron. So, sensory neuron, motor neuron is everywhere in the body,
Okay.
separated enough to shoot.
I can't.
Interneuron is difficult.
Even for like one neuron, my neuron tracker can't like keep it within like 1 micron.
Yeah, this okay. Go ahead.
So 50
Now I must
microns
realize.
is just enough to separate, for example, AVM and ALM.
Why can't we just express the protein in the one neuron we want to see?
One by one, yeah, it's okay, but then we have to
Yeah,
do that.
so-
302 different stuff.
So this is to the biology side. I'm not
It's
sure
just
how
for,
to.
like, interneurons, right?
Yeah, 100 interneuron. Maybe you have to make 100 different. You're right. We have to be realistic.
That'd be very
I don't think
laborious.
we can map all the neurons.
The only way to also
Okay.
do that while looking at some simulation is to use like a mesh instead of a channel obstacle, right?
Yeah.
Because as Charles said, you can't do both at the same time.
Right. So here is a specific.
Okay, so this is a small experiment that they performed using that system. So AVA is the interneuron,
Oh, I see.
which is
This
here.
is easy.
AVM and ALM are
So
both
separate.
mechanosensory neurons. And so they have channel reduction on the mechanosensory neurons, and they have GCAP and entry on the interneurons. So basically what they want to do is stimulate AVM and or ALM and see how it will affect AVA. And
So for the guy doing phototaxi, it's similar, basically. This guy is basically same as touching the body and the scale go backward, right? More or less,
Yeah,
that's what they
yeah.
found. So if it's the blue light, it goes to ASJ and it goes to AVA also. The same circuit.
so basically, yeah, they find that when the worm goes backward, the AVA has increase in calcium level. So this is a movie. This is not yet optogenetics, it's just a calcium imaging to kind of show
Oh,
you.
this is because 10x is not that great.
Yeah, so it's kind
Not
of,
much different from Steve's movie.
yeah, so it kind of shows you where AVA, ALM, AVM are.
We can do this one.
But the pattern is created the same way as you would do for
I
optogenetics.
also need somebody to make movies like that.
Now next is
Okay, so here will be optogenetics. So what you will see is
Hey, wait, they talk my idea. You
well,
said
it has GCAP and actually with optogenetics. Behavior has a beam on top.
you said yellow
okay,
circles go.
okay.
so this is where the AVA will.
Turn off the light. This is kind of fun. This is a video, right?
Yeah, this is a video.
Watch this video.
So this part is where AVA is. So you tag it with entry to kind of localize it, and GCAMP will tell you the activity of the neurons.
Right.
And then with optogenetics, we will shine somewhere, somewhere in here, and you will see that the activity of GCAMP will rise, and that the worm will go backward at time zero.
Okay. So
Oh, where are the brakes?
where it's the zero. That's
Yeah,
yeah, just
start
time
this.
equals 0 second.
So
Now it's on. With the backward.
yeah.
Omega. Was it Omega?
I think kind of.
This is the AVA activity. AVA is 2 seconds after shooting.
So they
Ah,
shoot it for
wait a second.
2.7.
That's 2 seconds, just like my system.
You're right.
Yeah.
That's like the
It takes 2
HJ.
seconds.
Yeah.
For
So
AVA to go
2.7.
backward.
No, that's just like the. I think it's something similar with like these fluorescence proteins.
This takes 2 seconds. That's interesting.
I'll-
No, it's just the YC3.60.
No, but it's not chameleon.
It's not the actual neuron that's taking that has latency. It's just the like the imaging, the fluorescence
It's
imaging. has
it's a
so
fluorescence
slow.
protein.
Yeah, it's just like it's a feature of the fluorescence
fluorescence
protein that they're
protein.
using.
G-camp is 2 seconds.
No, chameleon. No,
Chameleon.
This one is G-camp 3.
This one is G.
this
Okay,
is
but like
G-camp.
when he's talking, yeah, it's a similar concept between the two.
G-camp is much faster. I think.
Then what?
I don't think it's delayed by 2 seconds.
Somebody should read a paper on this. Look it up.
So we're talking about this delay of
There
2
has to be,
seconds,
like, the
whether
G-camp,
it is
G-camp works with, like, once
fluorescence
one
dye
function is triggered.
delaying
Yes, second
response
messenger.
or
There's second
actual
messenger, so
response
there's
of
going to be that
ABA
delay.
Yeah.
in 2 seconds after phototaxi.
I'm trying
That
to
was
just like constantly
Okay.
fix
No.
it.
Could you tell me that, right?
The chameleon.
So you get the idea basically instead of shooting a blue light
Not
for
like, it's not like a
photoreceptor,
It's just
you are
like,
shooting
it's not like the
into
behavior.
a touch receptor which is a
What they're
photoreceptor.
using.
Right.
Is like the,
Okay, so yeah, so this is the time where we should we stimulate the mechanosensory neuron, ALM or ABM, and then this is the activity of ABA and its correlation with the velocity of the
What is
body.
the wave velocity? Definition of wave velocity.
Wave velocity is just the velocity of the worm. Motion velocity.
Motion?
Yeah.
Ah, okay. So it's a body length is like a 0.1 millimeter.
Yeah, I got
Well,
kind of
this unit
confused
is
with
strange.
this unit too.
This unit is reversed.
Yeah, I don't know why they.
That's a strange unit.
What
It's
is?
second per
Yeah, so-
millimeter.
Yeah, I got confused.
Well, this is opposite. It should be millimeter per second.
Mhm.
No, it's body length.
It's too late.
If this is
No, but
body
this
length
is-
with the millimeter divided by second, it makes sense. It's 0.3.
Mhm, yeah.
I think this is mistype.
Yeah, it is mistyped,
so too.
yeah.
I think it's typo. And this goes to reversal.
Yeah.
Unless they actually do
And
it
so-
that way.
Now, I wonder this slight delay. This go reversal before this ABA is flashed.
I mean,
It looks
if
like
ABA is
change
like,
of velocity comes first,
because if
ABA
ABA
comes
is a forward
next.
facing,
Yeah.
exactly, like, when
So they also do a
that makes sense.
correlation
If it's reversing,
for
that's going to take some time
ABA
before-
activity amplitude and
some
the
other
velocity
neuron
of
would be activated, right? Not
the
ABA.
worm, and
Or
they
am I-
see some
no?
correlation. So finally, this is my end of my presentation. I want to talk a little bit about the data analysis for this kind of experiment. So far, our analysis group has been great in doing analysis of the motion of the body, but now we have to kind of think about how to analyze neuron activity, and this is very hard. So last year, for the past 2 quarters, I was in France, and
Oh, you are running this?
So yeah, so I had to treat some image of a zebrafish.
Oh, you are
live
working on
cells.
zebrafish of
Yeah.
fluorescence image.
Yeah. So and then basically what I have to do is quantify the neural activity, glial cells activity.
Oh, how many neurons you are watching? 1,000?
Not 1,000. I would say 50, something like
50?
this. Yeah. not that
G-camp?
Yeah. So basically, so this is a very long process. I mean, I was alone, and the data analysis group are not
But it's
much
immobilized,
better.
right?
Yeah. So they should be better than me. But
It's worse than it's
basically-
moving.
Yeah, so basically the hardest part in data analysis of neural activity is image segmentation. So believe it or not, what they did in this experiment is that they went to each frame and selected the neuron that they want
One
to
by
use.
one by eyes?
One by one, yeah.
That's what you are doing in
No,
France? Circle, circle,
yeah.
circle.
So yeah, this takes a very long time.
We have enough manpower.
Yeah,
Yeah.
so yeah, so
Oh, yeah.
what you might want to do is try to automate this image segmentation process. There are some code that do that.
We can do
By the way, do you know
triangulation.
what was this room 20, 30 years ago? This was for bulk chamber scanning. All days in particle physics, you take an image of 100 particles taken by film. And then we have 20, 30 women sitting here and scan an image one by one to find a new particle. I'm not joking. And there was a computer here. So this is the bubble chamber analysis room 20 years ago. Before I came here, more than 30 years ago. All days, there was no automated data taking. We have to rely on women because many patients in that. So we hired 20, 30 patients. This was a crazy operation 30 years ago. And for example, the famous one is the discovery of omega particle. It happens 1 out of 10,000 films. So you have to find one pattern, special pattern, out of 10,000 films to discover one particle. Omega is the bound state of 3 strange codes, by the way. That was discovered by scanning millions, not millions, 10,000, 100,000 films. So we can do it.
So you like just select on graph.
Yeah, and so once you have the image segmentation of the cell of the neuron, then you want to have signal treatment. And so you want, one common way to do it is to do the fluorescence.
Fluorescence change over the initial.
But again, as I said, your zebrafish immobilized.
Yeah,
Right? If we have a serious moving around,
No.
it's
That's
much difficult.
it's been more difficult. And for me, it was already very difficult. So
No, it's because like
You
I don't know. I'm working as like chameleon stuff for like a year, so like I know all about that stuff.
And you're watching only one. We are going to
Yeah.
have 302 neurons
yeah. And
in a
so,
couple months.
Yeah, so for this delta F over F naught, there are also many ways to do the calculation depending on the cell, because each cells have different like calcium onset and stuff like this. And then finally, maybe you can also do a raster plot to see the cross correlation between neurons. Not only
Did you
to
do
see-
any principal component analysis? to find-
Yeah, I actually did that for image segmentation. This was the big part for image segmentation.
What was the frame rate? This was
One-
three-dimensional observation.
one volume.
Yeah, it was very slow. I remember
One volume per
it's
second.
like
or whatever.
3 FPS, I think.
And
So-
of course, zebrafish has action potential. So unless you operate like 100 frames per second, you are so
Yeah.
sparse in time.
But
right?
yeah, I didn't do it on neurons. I was doing data analysis on glial cells, and they have much slower
Slower
activities.
Clear cell.
Glial cells. Yeah.
It's clear.
Did you stimulate by something?
No, I didn't do any experiment. I just worked on
just
the
analyzed
computation.
it.
Yeah.
Okay. Thank you. So, Jimbo, I asked him, he is the more engineer side. Which one is this one?
They want me to.
So he's going to talk about how to utilize this DMD. That's what I asked.
Yeah, I think Charles has covered most of the setup of the system, and this is my first time to give a presentation with this thing. Yeah. And now that we know that we want to stimulate each neuron one by one, so one of the options is to use a DMD. A DMD is short for digital micro neural device, and it's like a spectral light modulator. And if we zoom in, we can see, we watch closer, and there are arid micro mirrors. And how it works, with each mirror, it is acting like a pixel. So with a two-dimensional array, we can generate a certain pattern with the on and off state. So we can see this is the illustration of a mirror, and when it's on state, it reflects the light to the projection lens, and the light come out. And when it's on off state, the light is reflected to light absorber, and it's off state. And the mirror is controlled by the electrostatic attraction by the electrodes. There are two electrodes, and this is one single mirror.
Unbelievable. This is like a 10 micron square.
Yeah, yeah.
Unbelievable. It's quite
Yes,
complicated.
yes. It's very small. And under the mirror, there's a CMOS memory cell that stores the state of the mirror. And once you load the state of the, once you load the information into the CMOS memory, it doesn't change the mirror state at once. And there's a need for a clock signal. It's called a mirror clocking pulse. And when the signal arrives, it will transfer the state of the mirror to the state of the, the state of the CMOS memory to the state of the mirror, so that maybe we can change them simultaneously.
So it's basically on and off. You can't control the intensity.
To control the intensity, you have to modulate it at a very high speed. It's like a pulse with modulation. Yeah.
How fast can you modulate?
It's very fast. It's about, for example, for a binary pattern, it can operate at about 30,000 times. 30,000 times. So if you want to generate
like analog signal. That's
if you want to generate a pattern with a grayscale, it might be about 25 frames per second. It will be slower because you have to move it very fast.
I see. So then that's like 100 times slower.
Yeah.
Okay.
If it is brighter, so most of the time it's in the on state.
Right.
If you want to make it darker, so
more off.
Yeah,
So
more.
it's not a sine wave.
No, so
off like this.
Yeah.
no, so basically you kind of have like 8 bits of information. If it's like, if they update like 30,000 times a second, and you have like a grayscale of 25 FPS, then that corresponds to like 8 bits of information.
Yeah.
Which is grayscale. Okay. Wow.
And, so in practice, for example, this is 768 times 1024 pixels, and this is probably the size we are going to order. And they are divided into blocks, such as here we have 16 blocks. And there are many modes of operation. I'm not going to describe them in detail. I'm just going to show one single block mode just to show how it works. So we can see in the blue bar, it shows the data is loading into the CMOS memory. And when the loading is completed, there will be a micro clocking pulse to transfer the state of the CMOS to the state of the mirror. And you can see that we have finished one block's loading. And block by block, we have the full image loaded. And this is how we generate a pattern. And Aristocrat want me to cover this paper, but it is actually published earlier than that one introduced by Charles, so the system is much simpler. And I don't think it's necessary to cover this system. And they use the two types of light gated ion pump. If we shoot blue light on the neurons with this one, it will be excited, and if we shoot green or yellow light on the neurons with hollow, it will inhibit it. And this is a simple concept of optogenetics manipulation. And this is also the video attached to this paper, and it's quite similar to the one we just see. And maybe we just see one of them. Yeah. This is the same, and the blue part is where we are targeting at. And after we shoot it, there will be a reversal. Yeah, it's kind of the same.
On the left side?
The left side is actually.
So this is the very first application of the genetics to hearing us.
Yeah, in the
first step.
This one is shooting like about half the width, half the length of the worm.
But they didn't look at neurons. Calcium image.
It's
the behavior.
Yeah, it's just.
Could you show the left side again?
The numbers look like it's hard.
So basically the light goes more or less everywhere in the head regions.
Yeah. Actually, it is.
It's fast. It's within a second.
Yeah, I don't know why it's.
Show me again. I don't think it takes a second after you shoot the blue light.
Because.
Score.
It's less than a second for my eyes.
Yeah.
It's
And
over 7,000.
Maybe it's because it stimulates the whole area. Maybe. More neurons.
This is a light one mutant, by the way. Maybe they are stimulating ASJ neurons by phototaxis. You know, the concern is, of course, they have photoreceptor here in ASJ, like our photo avoidance behavior. This blue light may stimulate photoreceptor and to go backward. Of course. That's what
This is exactly the phototag.
So,
We can't let them know.
okay.
Go on. And what we are going to do is to add our the DMD system to our existing line confocal system, and we decided to put it here, yeah. And first we have two paths of light. One is for the exciting beam, and one is the inhibitory beam. And this is like a telescope system that expand the beam. And
Javier, you said you want to shoot 4.5 from the top because it goes downward, never go back. But now you're shooting 473 from here, going up.
Yeah.
Is that okay? I don't know.
We might have to bring the 405 down actually. We couldn't expand it more than like 300 microns. So,
Oh, that's a different question.
But yeah, that's the only place you can put it because you have to see the entire worm and you can't see it from the 40.
If you want to shoot the body for more time, yes. If you shoot interviewer, maybe you can shoot from the top. I think yes. We should think about how to organize so many wavelengths.
yeah, maybe
Look at this. This has 5 wavelengths, right? 4, 5, 488, 473, 561, and 1490. This is a crazy system. Nobody has ever made this kind of crazy microscope with 5 different wavelengths shooting.
And the diaphragm will be here in the path.
Okay.
So, and here's some other information, and this is probably the one we might want to order. And the software control is have open source.
But I said if you're sure we need this one, purchase it immediately.
Okay. I will talk
Yeah.
to my.
Don't mess with the cheaper one we bought 2 years ago.
Okay. Thanks. Yeah, and I think the software part is rather difficult. I think it will be more difficult to.
You'll probably find a lab view. Pretty sure there's a.
This one has a level view.
No, probably C to B.
Anyone software guy interested in programming this device? We need more help. I need one software engineer to program
I
software,
think to
Anyway,
know.
think about that. Anyone interested here? I need a programmer.
Yeah, because you have to tell the
Right.
the position of the neurons.
More software
Yeah.
oriented projects.
It has to be more precise.
It's,
Yeah. And here's our future plans.
Okay.
Or in the end, specified parameters.
We have to move fast. We have only 2 months.
Okay, okay.
No.
Yeah.
You're tired here.
Yeah,
It's only 2 months.
Yeah, yes.
This project takes 6 months. We are not moving fast enough.
Yes.
So we have to try to move fast.
Yeah, yes. Yeah. And that's all for my presentation. And at last, I want to say a huge hello to everyone here. This is my name. I'm a major in optical engineering, and I will spend 2 months in UCLA. And here's a little about my research I've done before. And the first is about
Spectral
imaging. Yeah.
Sounds fancy. What is
we use light to scan over a 2-dimensional surface, like some sample. It's a mixed supporter, and we use principal component analysis to tell if how much of one exact component is. And the other thing I do is liquid sensor on 400 crystal cavity, but I'm still working on it. Yeah, and last, it's very good to be able to working with you guys. You guys are brilliant. Thank you very much.
Thank you. Fantastic. Just a foreign student came here 2 weeks ago. Your English is amazing. When I was hired at UCLA, at the faculty meeting, people seriously discussed whether this guy can teach or not. I'm not joking. When I came here 30 years, I couldn't speak English like he. And there was a big discussion. This guy can do a research, but this guy may never teach physics. To the end, after 30 years, I got a teaching award 10 times. So it worked. Your English is really good. Impressive. So, the bottom present. Here, present.
Oh, present. Hello everyone, so I'm sure that at this point in time, a lot of you are confused as to what optogenetics is actually all about. And I'm very excited about this technique because optogenetics is one of those really groundbreaking discoveries that only come once every 100 years or so. And it's actually been discovered by this man, or pioneered by this man, Carl Dizeroff of Stanford University.
This guy's crazy. He also
This guy's absolutely
invented.
insane.
You know, he didn't really invent, but he's so quick to sum up invention when it was still talking around.
That's why you don't want to. Professors about
He also is considered to be an inventor of clarity, transparent frame.
I don't think he invented that. I think he stole it from someone.
He also stole from someone,
Probably.
from my opinion. I'm sure he's going to get a Nobel Prize this year, next year.
Right, so 2016 Nobel Prize winner, probably a hopeful Nobel Prize
He already
winner.
got a $3 million or $1 million, I forgot.
Oh, from the
Challenging, right?
the Breakthrough Prize. I believe.
Breakthrough Prize.
Yes,
That
from
is
Mark
probably
Zuckerberg.
better.
Absolutely.
It's $1 million or $3 million.
Right, and that's why I chose a very bad picture of him because he's a bad
This
man.
guy is smart, probably smart.
Yeah, he's probably very intelligent. He went to Oxford University as well, so that's very cool.
So, Rachel, do you know this guy?
He's your professor.
Meet him and tell me your impression about this stuff.
Yeah, so optogenetics is one of those techniques that are it's really incredible, really. But in order to truly appreciate the significance of optogenetics and to truly appreciate what it's all about, you have to know a lot about molecular biology, which is why I want to kind of have a lecture on molecular
It's why I
biology.
asked you to present.
Oh, okay.
I don't know much.
Alrighty. So we need to learn all about proteins because this is what the technique is founded upon. You need to manipulate proteins. You need to find out ways to bring ions into your neuron, find out ways to depolarize your neuron, and all of that stuff. I'm sure a lot of you are confused as to how neurons actually communicate, and so I'll be giving a lecture on that this Friday or next Friday or so. So first, here are a diverse variety of proteins. This is my favorite protein, ATP synthase, very beautiful protein. It's an actual machine that's very cool. And to truly appreciate proteins, we need to learn about their building blocks. But before that, going back to optogenetics, like how does this all relate back to optogenetics? Well, the whole technique is founded upon or based upon the principle that you manipulate a certain protein on your cellular membrane, and that allows the extracellular gradient of your cell medium to actually allow the cell to open up its channels and allow these ions to flow through. But more on that a little bit later, because I know a lot of you are a little bit confused about that. So I'll just keep going. So here's a very prototypical amino acid. You have a carboxylic acid group here, amino group here. You don't really have to know that, but just know that the basic principle of how all of these things work is positive and negative charges. If there's any key takeaway you should take from this lecture, it's all things in nature are based upon Coulomb's law. It's a Coulombic interaction between positive and negative charges, and that's how we get so much complexity from these very simple molecules. So these are the building blocks of proteins, and life uses about 20 of them. And 8 of them, we, as humans, we need to gain from other sources like food. But these are the amino acid structures that are present, and these in combination will produce all the proteins in your body. So this is the ribosome.
21.
So this was 21, but
this one.
No,
it's
that's
like a blank. It's just a blank slate, right? It's 21 circles. Right. But this is the ribosome. So inside your body, you've probably heard in your typical introductory biology classes that you have a typical cell, and then inside your cell you have these things called ribosomes that produce proteins. But the way it works is it's a 3D printer. You have a strand of DNA, and a different mechanism will actually use that DNA to create a new template called RNA. And that RNA will be then fed into the ribosome to actually 3D print a protein using amino acid as building blocks. So you have different other sequences that hold these amino acids. And these amino acids will string together to form a long string of protein, and that's called the primary structure of a protein. So we'll move on. So this is the primary structure of a protein. It's just a long strand of amino acids. And different amino acids have different properties. Like if you notice this diagram over here, some are called aliphatic, some are called aromatic, some are called acidic, basic, hydrolyxic, sulfur-containing, amidic. We have a lot of different various kinds. But basically, it all boils down to the idea that through these different types of interactions, these amino acids can have different effects. And you can imagine that if you ever played with magnets as a kid, positive attracts negative, and so they're gonna kind of stick together like that. So if you can imagine this string of beads as a string of randomly combined positive and negative charges, these positive and negative charges can then interact with the environment and with itself to produce form and structure. So this is protein folding. This is what's known as a ribbon diagram. You'll see this very often in your upper division classes or other such classes. But it's basically a representation of the basic structure the protein can undergo. This is called an alpha helix. These are called beta sheets. They're just different motifs that these proteins can undertake. So it turns out that from one string of proteins, depending on your environment, you can have a whole multitude of combinations for how it can develop its final structure. And based on the most favorable form entropically, it will adopt the final structure in your body. So you can imagine that through a sequence of just chance and where the protein has actually been situated, it can produce a very novel or a very useful type of machine. And using that form, this protein can then adapt itself to different environments. So in this case, this protein goes through the hydrophilic and hydrophobic regions of the cell membrane. And so on the outside, it's mostly hydrophilic, interacting with the water solvents and interacting with other ions on the inside. And here we have the hydrophobic residues that are interacting with the hydrophobic tails of this membrane. So proteins are really cool because they can actually change in response to different stimuli. As you can see here, there's like an open and closed form of this particular protein, open and closed form, open and closed form. And sometimes proteins can interact with different chemicals, like there can be a ligand or a little molecule that can interact with the protein, and it changes shape in response to that. And the best analogy I could think of to proteins would be, I'm not sure if you guys are familiar, but there are these toys called bakugans developed in Japan, where you have this magnetic card, and if you throw this ball at it, it will change shape. And here's an actual cartoon representation of that. I think this is really cool because this is exactly what proteins are. They are little shapes that change shape in response to stimuli. And of course, if you were to put any other stimuli near this ball, it's not going to react because it's not specific to that protein. So every protein has a specific ligand or a specific chemical stimulus that it can interact with. It can be positive or negative charges, it can be light, it can be sound, it can be a whole multitude of different stimuli. So how does optogenetics work, right? Well, first of all, we learned that you form proteins from DNA. It's the central dogma of biology. It's the main tenet of how biology works. You have DNA. DNA will be transcribed into RNA. RNA will be translated into protein. And protein is the main basis for how this technique will work. So let me skip around really quickly. But what does changing the shape of the protein with light actually do, right? And that's a really good question. So if you have a tub of water with some food coloring, initially you're going to have a very dense area here with a lot of color, but eventually it's all going to diffuse and be a uniform distribution of this coloring agent. And inside your body, you have different concentrations of ions, of chemical charges, that are balanced by a multitude of different forces. So inside the cell, you're going to have a lot of anions, you're going to have a lot of potassium, you're going to have more chlorine on the outside of the cell, and you're going to have more sodium on the outside of the cell. So you can imagine that because the cell has this barrier with these different proteins all around it, you are going to allow some things to come in and some things not to come in. You're going to form a selective barrier to certain ions and certain proteins. And so if you were to then open up these channels somehow, you will then allow this sodium ion to flow in, this chlorine ion to flow in, potassium to flow out, and ion ions to flow out because that's what they want to do. Those are called electrical driving forces and chemical driving forces, and we call those the electrochemical gradients, right? So everything wants to reach a certain equilibrium between a selective membrane, and that's what the cell essentially does. So using optogenetics, we are actually able to take a protein that's sensitive to light from algae, from blue algae, and take that protein, somehow squeeze it into the DNA, the genome of the particular organism, express that protein on the surfaces of the neurons, and turn on the neurons via our own volition by shining light upon that neuron. And that's absolutely incredible. I think that's the most interesting thing that has ever come about in the past 20 years, because you are then able to essentially turn on a neuron whenever you want. And the way it works is, the way it works is these ions will then flow across their natural concentration gradients. And of course, here are some of the ions kind of flowing out of this particular channel. This channel is selective to potassium. The way it's actually structured will only fit potassium and nothing else. And so it's really interesting how there are selective barriers to certain ions here. But it will do something called an action potential. The way it works is, here you have positive charges on the top and negative charges on the bottom. The more positive you get, the more depolarized you are. Polarization meaning more negative, so you're going away from positive, so depolarization. Or hyperpolarization meaning more negative, going more towards the negative charges. So it turns out that when you open up these channels using optogenetics, these channels called channel rhodopsins, using light, you're allowing sodium to be permeable to the membrane. And sodium has a natural chemical gradient, natural equilibrium potential close to about positive 40 millivolts, which allows sodium to flow in. But because of the natural shape of these proteins, they kind of close after a while. They open, they close, they open and close. And stochastically, if you reach a certain value, they will keep opening, and just by chance, you'll open up all of these sodium channels. And because you have a lot more holes through your membrane, you're going to have a lot more ions flowing in, which causes this massive inflow of charge. And after a while, the same channel will then close, and another channel will open called potassium channels. They're voltage-gated potassium channels, and these are a lot slower to open. So while these sodium ions are actually flowing in, these potassium ion channels are also opening. But after around here, these potassium ions will then be at their maximum conductance and allow a lot of potassium ions to flow out of the
Well,
cell.
you are missing calcium.
Calcium
After
does not.
sodium, it's calcium, right?
Right. So this would be for a human neuron, but
also
for
have calcium following the sodium.
right, calcium is not like a significant player in the action potential. It does not play like a huge role for the actual depolarization of the membrane.
How would the calcium come after sodium in the action potential?
So calcium would be a secondary messenger
I know.
that helps to open up more channels for the cell. It does not actually.
It's not a part of this voltage change?
Not a part of the voltage change. It would be more of a.
synaptic clamps. It's involved in the synaptic clamps. I'll cover that part.
Right. So
The reason why I'm asking, of course, this fluorescence measurement is sensitive to calcium,
Yep.
not
So
sodium.
it turns out that for humans, we have sodium as our primary action potential driver. But for C. elegans, they don't. I guess they didn't.
C. elegans,
develop-
we are lucky because calcium is a primary.
Right. It's the primary
But they
driver.
don't have sodium. Yeah.
Right.
So the actual mechanism is very similar actually for C. elegans, because the way it works is you have a different protein that's very similar to the sodium channels, but it's also more permeable to calcium. And so in response to a certain voltage threshold, it will allow calcium to flow in, because calcium would be the predominant.
And the other question, maybe just a question to you, is we are talking about how quickly this ion flows. The current model has a time constant of 100 milliseconds, which is
For
very
chemical synapses.
And then electrical synapse is always faster.
Oh, so these would actually be gap junctions. So,
gap junction is
These
very
are
fast,
gap
right?
junctions.
yeah.
But action potential is not a gap junction.
Right. Oh, yeah, sorry.
So it comes from some synaptic connection to show me action potential is only a few milliseconds.
yeah.
So it can be triggered by synaptic connection too.
Yes.
We had discussion this morning with a simulation guy. They are producing like 100 millisecond order of rising and falling,
Wrong then, right?
which I think is wrong. It's too slow.
So that is very slow. So in this case, this entire action potential happens for a course of about 5 milliseconds.
Right.
That's for people though, like mammals. But for C. elegans, it's slower, no?
That's my question. I think it's not that slow, like 100
That's
milliseconds.
not about that in the slide.
I think it's like six.
So-
I think the EFIL paper said that like maybe like 60 milliseconds, like because that's like
Okay.
electrical response.
That makes more sense because calcium is
Yeah.
not very abundant in cells, and so it might
Yeah.
take a longer time for it to reach
All right.
full.
I need to know the answer.
Right.
Yeah.
If it's 60 milliseconds, it makes sense that this the modeling has 100 millisecond time response.
Yeah, I think
This one is so fast.
Absolutely.
I think like yeah.
All right. So
But the
let's
reason-
find out in a few days.
Right.
I want to know the answer.
Yes.
Few milliseconds or 100 milliseconds is a big difference.
Yeah, that's
Okay.
true. Order of magnitude.
But the reason why it's so fast is because you're opening up a lot of these channels, and sodium just has a giant electrochemical force. So here's the driving force.
Calcium is not.
The calcium
It has a
because
large-
it's very minute concentration. You have a very small concentration, so very small chemical driving force, and so that's going to cause a
Yeah.
slower
And
propagation
like
of
the
voltage change.
the gradient
But-
between like the inside and outside of the cell.
Yeah, it's a
is
beautiful,
less.
beautiful way of
So it's, like,
actually
less.
transmitting information because I'll get more into the actual transmission of information this Friday or next Friday, but that is the basics
By the way,
of action.
Voltage sensing fluorescence dye is a big industry now.
Yes.
Nobody really succeeded by doing so in genetic way.
Yes.
We have to bump it. I did it once. It's not easy. But if someone invent the way to genetically encode voltage sensing fluorescence dye, you see this potential immediately by fluorescence. We don't even need to look at sodium ion. And that is the way to go. I don't know when it happens. But once it happens, it completely changes how to do a neuroscience. You can see this pattern by fluorescence
Talking
signal.
about calcium indicators, right?
No, not calcium, I'm talking about voltage sensing.
Voltage sensor.
So someone may want to look into that.
No, so then it's a direct signal of when the
Yeah.
neuron activates.
Absolutely.
Interesting, interesting.
It's the most. It's like electrophysiology.
It
really is
It's
like
right. It's electrophysiology. It's the
Yeah.
same as patch clapping doing by
Right.
Fluorescent patch
fluorescence.
clogging, Yeah.
Absolutely. So yeah, that's optogenetics, and that's why it's so powerful because you can manipulate these neurons by simply opening up these channels. Now, this protein for optogenetics, this rhodopsin, is sensitive to light. We talked about how different proteins have sensitivities to different chemical stimuli or different stimuli, but this particular protein derived from blue algae are particularly sensitive to blue light, and so we can open these channels, allow sodium to flow in, cause the depolarization or the action potential, which is basically a signal for an active neuron. And so we'll talk about why that is a signal for an active neuron for another talk. But that's optogenetics, and that's why it's so powerful. Thank you.
Thank you. Okay, it's a little bit late, let's finish. The last talk, it's kind of fun. I'm running something. So, one more talk and that's it about optogenetics. Where is it? Here. Which talk? This one?
Yeah.
PDF files?
Yeah.
This is the PDF. Oh, before reading here.
Disclaimer, I'm not going to read everything on the slides, but just work along with me. Okay, so I'm going to cover Mikey's lecture in a bit more detail. So we've noticed that, research has kind of crescendoed towards this realm of non-invasive investigation, shining light on neurons to activate them without surgical, invasion. And that's a really important motif that we've observed in the community of C. elegans today. So let's talk about optogenetics in a little bit more detail. Well, basically, it's activation or inhibition of neurons based on expression of a protein on the receptor surface of the neuron. So C. elegans are a very streamlined, accessible model for this because we have full delineation of their connectome, we understand how optically accessible they are, and we have full quantification of their behaviors. So stimulate this neuron, maybe they'll do omega turn, and so on. So we've dealt with two protein receptors on the surface so far, channelrhodopsin and halorhodopsin, which have been expressed, most prominently in C. elegans, and it's continued so forth. So before we even discuss that, we need to talk about gap junctions, which are constituted by 2 connexons, and each connexon is constituted by 6 connexins. So basic form of facilitation between cellular components. We got ions and other particles that need to migrate between cells, so this sort of gives them that facilitated motility that they need in order to move from one cell to another. Now, gap junctions in the simplest form of neurons are synaptic clefts. So if we take a look at chemical synapses in neurons, we have a very simple model that involves calcium. This is a very similar model to how C. elegans function in a lot of other mammals and more multicellular organisms. Basically, you have an action potential that travels down the axon terminal. Depolarization causes calcium channels to open up. An influx of them will basically lead to exocytosis, which means vesicular fusion of these little membranes, these little circles right here, to the presynaptic membrane, and that's what basically extrudes the neurotransmitters. That's what spits them out into the synaptic cleft. Now that's important because C. elegans have 2 very prominent neurotransmitters. One is GABA and the other one is acetylcholine. They have very adverse effects. GABA is usually inhibitory and acetylcholine is excitatory. Now, the major type of GABAergic cells in neurons are inhibitory, and then acetylcholine-based cells are excitatory. So if you take a look at the the full delineation of connections, the synapses will usually fall into 3 categories. They could be monadic, dyadic, or triadic, and that means how does the neuron interface with the successive neuron, and how many of those successive neurons are aligning around it. So we have around 6,300 chemical synapses and 800 rough, chemical junctions, electrical junctions, and then 1,400 neuromuscular junctions. So that should give a scale. Of the 302 neurons, we have so many more permutations of interfacing involved in that regard. Now, if we go back, just to cover the process that Mikey discussed with action potentials, yes, calcium is a very significant player in C. elegans. They don't possess the conventional sodium ions that we're so used to hearing about when there is depolarization events. But the resting potential in C. elegans is pretty similar to humans. I believe it's in the -70 range for, is it volts, millivolts?
Millivolts.
Millivolts, yeah. Thank you, Steve. So the depolarization effectively causes -70 to move into a positive range, and that's where you have the influx of channels of calcium and other things that go inside the membrane of the cell. That's important because once your voltage becomes positive, you have the ability to do work. It's potential. And that potential is the impetus that drives your action potential from the axon terminal down to the dendrites of the successive neuron. So now that we've got that out of the way, let's talk about some of the neuromolecular processes and the interesting questions that they kind of exhume. So how can we mark ASJ, which is a neuron, with chameleon? Chameleon is a calcium indicator, and we've done this. We've utilized it for calcium imaging. How can we make QW1217, that specific strain, which we already have? How can we make AIY with channelrhodopsin, which Mikey kind of grazed on? And the overarching questions, how can we mark excitatory and inhibitory neurons with a fully streamlined and distinguishable method? And how can we mark all neurons with channelrhodopsin or halorhodopsin? So let's touch on that. So Mikey discussed, you extract this specific segment of DNA from green algae, I believe, green algae, and then you would use a plasmid vector to transfect it into the gonads, the specific region, the gonads of the C. elegans, and that allows for expression of this particular protein, a light photoreceptive protein channel. which is activated by blue light, or anything within the 430 to 460 nanometer range. When that happens, you open up the channel, you have an influx of ions, and that is the depolarization event that basically engenders the potential for action potentials to carry on forth. You need positive potential for that to happen. Okay, so fundamentally, what have we done so far in the realm of neural imaging? What have we done so far in the realm of ionic channels? Well, in this lab in particular, we've worked with GECI's chameleon. Steve has a lot of experience in that.
We haven't. What?
Okay, wait. No.
We have a GCAM, but we don't have a GECI.
No. I'm talking about chameleon. Chameleon, what Steve has been working on.
What GECI means?
Oh, these are genetically encoded calcium indicators. Yeah, same thing.
Genetically.
Yeah, genetically encoded.
It's like RFP, like anything we use in our lab.
Yeah, basically.
It's just a fancier name. I don't know.
Yeah, so these are basically accessible ways to visualize spatiotemporal activities, that fourth dimensional stuff, blah, blah. So you can employ color-shifted fluorescent probes, which means you can have red-shifted, you can have blue-shifted. Red-shifted is good for penetration in deeper tissues and all that stuff. So more importantly, we can take this type of ocularity that we have with this and integrate it into optogenetic experiments with photoactivable proteins. So now you have calcium indicators that once they bind to the protein receptors, the fluorescence will basically turn on or off, and then you can indicate what's happening. Is the neuron being activated or deactivated? So these are just some food for thought. What can we do to distinguish excitable versus inhibitory neurons? Because we know that the C. elegans has this sort of symmetry, and it works with both excitatory and inhibitory neurons. So here's some contemporary research that could potentially be utilized. In situ patch clamping, so it's electrophysiological technique which can keep the current constant around the protein channel, which means the cell no longer relies on ion currents, it relies on a constant voltage. And by utilizing that constant voltage, you can basically debunk voltage current relationships. And excitatory neurons will always have more voltage-gated channels than inhibitory neurons. So just by assessing that relationship alone, you can identify excitatory versus inhibitory given a fluorescent tag or anything of that sort. Antibody staining. We have antibodies available for GABA receptors and acetylcholine, so that's very accessible in the C. elegans as well. GABA is generally an inhibitory neurotransmitter. Acetylcholine is a excitatory neurotransmitter. And then we have something that was suggested by Blake, which is viral injection of fluorescent components. The virus will bind to dopamine receptors on the surface of particular neurons, and when that happens, they can inject their genetic material, which contains the fluorescent components. But they'll only have affinity for specific receptors on neurons. So if we can basically curtail that to specific neurotransmitters and neurons, then we can delineate this neuron, this neuron, this neuron, whether it's excitatory, inhibitory, and blah, blah, blah.
It only works for dopamine?
No, it can be utilized in extension. That was what it was used with
So
first.
let's assume, according to this, ideally, I want to mark GABA receptor and acetylcholine receptor separately.
Okay.
And take a static image. Can we do it?
So what would happen is you would need to manufacture a virus that has affinity for the receptor specifically, and then it would inject a viral component.
Has anyone died, either mouse or any animal?
Oh, they've done it with mice.
Mice?
So it's
So
pretty
there is
accessible.
a mice experiment. Someone already expressed GABA receptor by some fluorescence dyes.
No, I have not read on that yet.
So I think
know.
it's two questions. One is whether, as a static image, like a connectome, can we mark only GABA receptor by GFP something, and scan slowly?
Right. Think about it though. All sorts of neurons contain both GABA and acetylcholine receptors. So if you mark one, it doesn't necessarily distinguish excitatory.
But if you have extremely good resolution to pinpoint the location of every single synapse, can we tell?
Can we tell?
If we have a super resolution microscope and I can tell this is the synapse from neuron A to B, and look at the distribution of the GABA. And if the microscope is high resolution, can we do this?
Can we do
that? Wouldn't be necessary because all you need to really do is activate all neurons are associated with that synaptic potential and see whether the output would be.
But that is optogenetics, so active measurement. It's the second question. But first question, can we do by static image? Because there is a gamma
With viral
receptor.
affinity.
We don't have to activate if you can.
I'm pretty
identify.
sure this was a static image
Yeah.
observation.
Right. That's what Blake
Yeah.
was telling, right?
Yeah, that
for
was
dopamine.
This was a
So
static
for dopamine mouse, they did it.
Yes.
My question is, can we do the same technique for gamma receptor and acetylcholine
That's
receptor?
what Blake
in CNS?
suggested. What was the extension of this technique? Could it be used? It's food for thought.
Then that is easier than dynamic measurement of the genetics.
Yeah, naturally.
We just scan very slowly, high resolution, super resolution.
Right.
So and if it doesn't work, we go to optogenetics.
Yeah.
So we dynamically stimulate one neuron and see the propagation to the next neuron.
Yes, exactly. So on the topic of photoactivation in optogenetics, channelrhodopsin, activated by blue light. We've already discussed, CHR2 that has been expressed in the body walls or mechanosensory neurons of the C. elegans has actually initiated contraction in reverse for illumination of 430 to 460 nanometers. And we also see some mutations that they've basically unveiled that I believe induce stronger action potentials when you exchange the histidine which is the protein that Mikey was going through, the 20 amino acids. So histidine is exchanged at position 134 to an arginine residue that can actually enhance action potential, make it more robust. So this is something that our first speaker was discussing. The problem with optogenetics is that if you were to utilize CHR2 and then any other optically active
So if I do calcium imaging and optogenetics,
you can't. They would have overlapping excitation spectra, and so you couldn't be able to distinguish the signals necessarily. So you have to consider the excitation spectra of
You
it.
can't do it because of the rolling shutter.
GCAM3 with the you couldn't do that.
yeah.
Well, we
They're
don't know
overlapping
yet.
spectrum.
Wait, could you do it? Could you do it like this?
Like conceptually, it's possible.
Conceptually, because it's a time difference between them.
Conceptually,
So
yes.
you should really think about that. It's an innovation, right? Then we don't have to
What would
worry.
be the time margin between those two events, though?
This line is moving fast, 100 kilohertz order. So basically, this line is moving like 200 frames or 300 frames.
Well, I want to talk to you about that afterwards for
1,000
sure.
times.
1,000 times.
2,000. It's
Anyway,
like
this is
microsecond
line
order for like.
of the blue light
Yeah.
is moving as a function of time.
Right.
Now, let's assume you shoot a blue light for excitation of synchronizations, right?
Yes.
This beam does not is not recorded by camera because camera is reading only this line.
Oh, so if you synchronize
This
it
may be an
to.
innovation.
Oh wait,
Shoot in the downtime.
no. So okay. So like let's say.
The line itself is much
No.
faster than the actual beam that's being used for optogenetics.
You're right.
So.
This should work.
No, yeah. So if we have like a synchronization to within like 1 millisecond.
This
So
is
basically,
really interesting. Nobody has ever thought about that.
So yeah, basically the way it works is we don't image the whole worm all at once. We image only part of it.
Oh, we image line by line. at the
Line by
function
line,
time,
right.
Yeah.
So every single time we just open a slight slit.
Okay.
So here the ceiling. Let's just write down that everyone understands. I think it's going to work. You're right. It's smart. It's good to have a meeting. We invented new technique. So let's assume we are sending a line. Line. T equals 0, right? T equals 1 millisecond.
Yeah.
Every 1 millisecond we're slicing. The camera only reads this line at a given time. So while we are reading this line, we can shoot a beam here. This is not recorded. Camera is blind to your excitations by blue light for channel reduction.
The second camera records
The
that.
camera.
The second camera would record that.
Well, what I mean? Camera. What I mean?
But is
Camera.
it the overall?
He means the one
The
tracker
over-
one,
yeah.
the one that's not doing the signal.
Yeah.
This may work.
So then you could use blue light and excite both of these and then distinguish those two signals.
We can try with the photostimulation. Blue light, we basically off synchronize with the blue light out from.
So then I have to link my synchronization program with the.
So finish your line confocal anyway, right? It's not quite working. So once we establish this line confocal, we just off synchronize your blue light stimulations.
It might be
Shoot the
possible.
extreme blue, bright blue light, your camera is insensitive because you don't open the shutter.
Oh, we'll think about it. Yeah.
good. Let's good. Yeah,
This
let's
may
have
work
this
indeed.
discussion.
Yeah, maybe we can mix our G-camp.
Good for spawning new ideas. Next part, photo inhibition by halorhodopsin. So some people say green light, some people say yellow light. Really, it's up in that echelon of 560 to 590 nanometers. This is the protein receptor channel that will basically inhibit action potentials because it provides an influx of Cl minus. And Cl minus is going to repolarize the cell, which is bad for action potentials. So when it's expressed in muscles, the worm is basically paralyzed upon yellow light inundation. Okay, so limitations we discussed, but we just innovated a plan around that. That's really interesting. You also got to look at the kinetics of it. So sometimes it takes an order of 30 nanoseconds to a whole second just for the process of action potentials to be activated in successive neurons. So that's important for long-term articulations or long-term analysis of neuromuscular junctions and other things that happen. So consider those two: the protein's excitation spectra and the level of kinetic response when utilizing optogenetics. Now, this is what we talked about. We talked about expressing the protein in a single neuron. Now, the problem with that is that, if I skip forward, finding promoters for single cell expression is kind of difficult. Now, there is a method that was...
But that's not our direction, right?
Huh?
We try to shoot a specific neuron by a beam spot.
Right.
So it's okay to have many cells marked together.
Okay, but I guess just explaining the technique would never hurt.
So just to make sure that two ways to stimulate only one neuron.
Right.
One is only that neuron has channel rhodopsin attached and shoot a light everywhere. The second approach is to attach channel rhodopsin everywhere, but basically shoot only one neuron.
True.
That should be our approach, I think.
So you wouldn't want to...
So, yeah, don't worry too much, because we have to look 302 neuron anyway.
Yeah,
so
naturally.
even if it works, it's so time consuming to make 302 different neurons one by one, right?
Well, this would have just involved intersection of two promoters.
But at least for some neuron like
Yeah.
ASJ, AFD, AY, someone can do it, right?
Yeah.
We already have a sample of chameleon. So instead of chameleon, in principle, this guy can attach channel.
Okay, so then rather we would just hit one neuron at a time instead of
For
hitting
that specific one,
Yeah.
someone already know how to do ASJ, AFD, AY,
Okay.
which we have.
Yeah, then forget about this technique for now. Yeah, so let's take a look at a little bit of biopreparation. Well, you would need to grow the transgenic worm, the one transfected with the plasmid vector, with ATR. So that's cofactor all-trans-retinol. It's important for the photoactivated receptor on the surface. So optogenetic proteins expressed in C. elegans, they're activated.
So how do you do the virus injections? That's the question. What do you do with this manipulation of the gene? This is just a
How do you actually make the C. elegans sample like with the virus?
Yeah,
that's my question.
Okay, let me go back one slide. So basically, they utilized two methods. It was either microbombardment or actually injecting it into the gonads of the C. elegans, that specific region. They would take the plasmid vector, they'd put the DNA sequence of interest using restriction enzymes and then fitting complementary sticky ends. And then
what facility do we need?
Well, we just need some plasmid vectors, some restriction enzymes for that, and then we can shove it in.
Can you do it?
We can do it. This has been done before.
Actually, Tiffany told me that she worked with Elisa Helen before.
Oh, you worked with Elisa Helen?
No, I never.
Or she spoke with Elisa Helen.
You talked to Elisa
and
Helen?
Yeah.
According to her, you don't need to inject C. elegans. C. elegans can just ingest the food in the form of E. coli.
Oh, yes, we can.
That was
We can eat
That's RNA. I don't know if
we
it
can
works.
feed RNA.
This is
That's one way
different.
to do
This is a
that.
bit different.
Injection.
Yeah, this is different.
But you can still express. Okay.
Yeah, but once it's in the body, you would want to express that cofactor all-trans-retinol. So you would put that in the bacterial lawn when preparing it. It's very simple because we already have bacterial lawn with E. coli and other foods. So we just put the ATR in a small enough quantity.
So I'm sure if you go to the facility which Elisa Helen will use it, there is a facility, but we don't know how to use it, so we never approach. I don't know what to speak.
I can do some research into that.
From Japan.
Yeah, she knows how to use the system, so that she's coming in a couple of weeks indeed. She can teach us how to have a facility. So someone should understand principle and facility, and this as Japanese student can teach you how to do it in August.
Okay. Yeah. Okay. We went through basically everything you need to know for optogenetics at this level. And just take a look at one or two promoters. Once you have the transgenic C. elegans by injecting the plasmid into the worm's gonads or whatever other methods that you have proposed. We work with a few prominent promoters in the body. MEC4 promoter is really convenient when you want to drive expression for gentle touch stimulation. So we have the mechanosensory neurons. And then remember the eyelash experiments and other things like that. You'd be dealing with that realm of stuff. And then for inhibition, you can work with MYO3 or UNC17. So these are promoters that would drive expression in cholinergic.
The published paper based on this kind of stuff?
Yeah. This was all published. This is all contemporary research.
Okay, the other approach is to contact this guy and get a sample, as I do from Japan.
Okay.
And just to make sure, what I have done is first I contacted Samuel lab and got QW1217.
Right.
Next I, of course, Elisa Harlem gave original one like ST2 and AIY like that. And then I got ASJ from Japanese group. So this community of C. elegans is very friendly.
Right.
It looks like whoever you contact, they are supposed to give you a sample.
Right. And the motif of these approaches is basically they're trying to utilize protein.
So why don't you get genetic sample from this guy who already published with a motor neuron?
Yeah.
And try. It's easy. We have a blue light already. Shoot a blue light means the phototaxi. Shoot a blue light. It's 4-5, so wavelength is not correct. But anyway, motor neuron is easy. Or the sensory neuron, the touch sensor. It's so sparse, right? ALM or whatever. What is it?
There are six of these.
So
very sparse.
Let's try something rather than making our own sample first and practice simple optogenetics.
Okay. And really, they only discuss these because you utilize protein.
I don't think it's not that difficult because there are 20, 30 laboratories doing this already 5 years
it's
ago.
pretty accessible.
Right? So from our kind of
Facility.
technical skill, we should be able to do it in 2 months.
Okay. Well then, yeah, so I've covered basically every slide, but just understand that this is the direction that our current compendium of research is moving towards, non-invasive investigation. And so with optogenetics, we should be able to debunk a lot of these mysteries, a lot of these commonly inaccessible realms of information that C. elegans will hopefully enlighten and reveal for us.
Thank you.
So thank you guys. Yeah.
So it was a long presentation, but I have a much better idea. Now I realized all the optogenetics was done 4 or 5 years ago. For either motor neuron or touch sensory neuron, which is so sparse in the body. So it's easy to target sensory neuron or touch or motor neuron. But that isn't so exciting for me because it's a kind of reflex. Of course, if you touch it, it goes backward. Instead of touch, you excite by blue light, so what? Of course, it goes backward. Or motor neuron, if you hit the motor neuron, go to the left, it goes to the left. So that's what I saw today. And then, for the last 4 or 5 years, not much has changed. Because excitement comes if you shoot a blue light on one of the interneuron, not sensory neuron. Sensory neuron is identical of sending some sense signal, right? Motor neuron is so downstream, of course it works. In between is a difficult part, but it's dense. It's packed together like 100 neurons, packed every 10 micron. And you can't shoot one each, and you can't only mark one at one time, right?
So the technique behind you would work to basically target a specific neuron of expression. See, just like you said, you could shoot just one neuron, but the region where it's dense,
it's very dense.
Right here, they can find a way to decrease the subset of expression to only one cell based on this intersection of two promoters. Very rarely will neurons have this intersection of two promoters.
So first, of course, this Japanese friend can mark at ASJ or AIY instead chameleon. They can channel adoption for us, right?
Right.
Maybe I should ask them just to start.
But you're saying it'd be difficult to mark a single interneuron because the region is so dense.
But that's what we have. AIY or AFD maybe not that, but AIY is dense, right?
This technique right here, this technique right over here on this slide would allow you to even hit regions of incredible density, just because the permutations involved.
I think the difficulty is interneuron, so dense, and how to only excite one by one and see the propagation.
If you can see the protein expression of each of those neurons, just target it using CRISPR.
Yeah, but how efficient is it at selectively?
Because we have so many salients. We
Yeah.
can pull off the ones that are efficient.
Anyway, the meeting gets long. Yeah. Okay. So let's break, and you're welcome to take more data for the rest of the day, and let's keep talking about this exciting things.
Have you been able to
Thank you.
express